In this video, I'm going to be talking more and in depth and further about biofilms and onco-biofilms and how integrins and cholesterol loading in lipid rafts can actually play a role in biofilm formation. Now, recap on biofilms is that if they get too thick and they are not degraded, they can actually get thicker and much worse if, let's say, for example, a macrophage, which is an immune cell, attempts to engulf or phagocytose, or another name for phagocytosis, the immune cell tries to do that on a cancer cell or pathogen that is covered in a biofilm. So that could actually make things worse. That's why it's important to prevent the biofilms in the first place or to try to degrade the biofilm. Okay, so basically, lipid rafts are in the plasma membrane, rich in cholesterol, and they act as platforms for signaling proteins, including integrins. And high cholesterol can increase the stability and size of lipid rafts, promoting the clustering of integrins. So integrins, they actually strengthen cell adhesion. That's actually what biofilms need. They need high adhesion. And what promotes these integrins is clustering in the lipid rafts. So therefore, high cholesterol, more lipid raft integrin clustering, therefore leading to stronger adhesion that could enhance onco-biofilm formation for cancer cells, protecting cancer cells against immune attack. And this is due to cell-to-cell compaction and ECM deposition, like in EPS and microbial biofilms. Okay, now there are like experimental ways, such as with cyclodextrins. That's what scientists do to experiment with extracting or taking cholesterol out of the lipid rafts. But cyclodextrins cannot be found in diets in meaningful amounts. But altering the diet can change systemic cholesterol in the membrane, influencing rafts. Because rigid rafts, they promote integrins that allow adhesion for the biofilms. So some relevant nutrients include phytosterols, such as in nuts and seeds, and modest lowering in LDL and reduced intestinal cholesterol absorption. So theoretically, that could reduce the cholesterol that are making the lipid rafts rigid that are supporting the biofilms. Also, omega-3 fatty acids can disrupt ordered lipid domains in the rafts, therefore increasing membrane fluidity, reducing raft stability. Because that stability of the rafts is the reason biofilms are forming, and then also polyphenols, examples resveratrol and quercetin. Some studies suggest that may modulate raft signaling, affecting microbial biofilms, but evidence is still emerging. So some foods here that might help. So pistachios, key compounds, phytosterols, polyphenols, MUFAs, short for monounsaturated fats. The possible effects is LDL cholesterol reduction, uh, improved membrane lipid composition, mild reduction of raft cholesterol density. So that could mean that biofilms are less likely to form thanks to pistachios, because biofilms need rigid rafts, but pistachios and monounsaturated fats reduces those lipid rafts. Okay, as I said earlier, polyphenols can modulate raft signaling and affect microbial biofilms. And the next food or nut here on the list is pecans, which happens to have these polyphenols, being an anti-inflammatory and influencing raft signaling. So the next nut here is probably the best one here, or one, or at least one of them, and that is macadamia nuts. I've talked a lot about this in many of my other videos. Macadamia nuts have many anti-cancer benefits, and especially here, because very high in oleic acid and alters membrane fatty acid composition, increasing fluidity, therefore reducing raft rigidity, uh, reducing the integrin adhesion that biofilms rely or depend on in order to form. Now, macadamia nuts are critical for reducing biofilms because, again, uh, if biofilms don't break down, then the macrophage will try to engulf it, which will only make the biofilm worse and even thicker. The next one here, not as good as macadamia nuts, and that's peanuts. Peanuts, they do have these aflatoxins, like these molds that have grown on the peanuts, possibly during processing, even before like being bagged or turned into peanut butter. So peanuts could possibly be at the bottom of the list due to aflatoxin. The peanuts, they do have phytosterols and polyphenols, therefore possible mild or modest biofilm inhibition. But still, due to the aflatoxins, maybe not a good idea. Next one is Brazil nuts. Have to be careful with this one because very high in selenium. Selenium is needed for the thyroid. Too much cannot be good. And Brazil nuts, it does have an indirect biofilm influence, improving immune response in microbes. The next one is almonds, has phytosterols, so indirect raft modulation, therefore reducing the rigidity that integrins provides the adhesion for biofilms, also reduces LDL as well, and oxidative protection of membranes. Important because oxidative membranes could be the reason as to why lipid rafts are rigid and providing the integrins for the biofilms. Next is cashews, lower sterol content, so not as good as the other nuts and seeds because phytosterols, they do help reduce lipid rafts and integrins because phytosterols, they compete with cholesterol at the membrane. So cashews are not as good as the other nuts. Modest lipid effects. There's also less evidence on cashews when it comes to rafts, proving that cashews are not that great. Cashews, they do have magnesium though. That's important. Lysosomal membrane peroxidation, that the cathepsins can degrade the exosomes. Tumors cannot get the exosomes. Magnesium is also important to support potassium levels because potassium deficiency cannot be fixed if there is no magnesium. That's important to keep blood glucose out of the bloodstream by upregulating GLUT4 for the cells to take glucose out of the bloodstream. That's important because biofilms, they can upregulate VEGF, which is angiogenesis, which is blood vessels that give sugar or blood glucose to tumors as a fuel source to metastasize. The goal with potassium is to make sure that there is less blood glucose in the bloodstream so that the tumors cannot get the glucose through the blood vessels. Next is sunflower seeds, high in vitamin E, increases membrane fluidity, therefore less lipid raft clustering, less integrins, less adhesion for biofilms. Next is pumpkin seeds, high in zinc, as well as polyphenols and phytosterols. So, phytosterols will compete for cholesterol at the lipid rafts, reducing biofilms, and polyphenols also reduces biofilms. Zinc in pumpkin seeds is important to reduce MMP9 because MMP9 promotes invasive oncobiofilms. So that's it for the video. If you want more future videos, then like, comment, share, and subscribe. Keep it locked in here on my channel, and I'll see you in the next video.